Hi, I am Dr. Mike Van Tielen with NeoMatrix Medical, the most powerful name in regenerative medicine. Today we will discuss the 5 reasons not to use your own stem cells. Reason number 1, the amount or quantity of stem cells drastically declines in our body with age. Scientific research clearly shows that as we age, our body's ability to regenerate damaged tissue decreases. That's because the quantity and quality of the stem cells in our bodies begin to significantly diminish over time. In this graph, we can see that in a newborn, 1 out of 10,000 cells is a stem cell, and that number drastically declines with age. As a teenager, only 1 out of 100,000 cells is a stem cell, and when we're 50 or 60 years old, 1 out of 2 million cells is a stem cell. Reason number 2, the quality of stem cells in our body drastically declines with age. How do scientists explain that the quality of stem cells in our body drastically declines with age? Telomeres. Now, what are telomeres? Telomeres are the end caps that protect our chromosomes. And over time, telomeres shorten. And when they shorten, their ability to replicate and differentiate reduces. And eventually, cell division stops. We already know that a stem cell is different from any other cell in our body because a stem cell has 2 cardinal properties. Number 1, a stem cell can replicate itself. It can make perfect copies of itself upon division. And number 2, a stem cell can differentiate into many different cell types in the body. Now, as we age, the quality or the ability to replicate and differentiate drastically declines with age. The reason we recommend not to use your own stem cells is because the quantity and quality of the stem cells in our body drastically declines with age. Besides those 2 main reasons, there are more reasons. Reason number 3, the essential components for proper tissue engineering may be missing. For effective and consistent tissue repair, tissue renewal, and tissue engineering, the body needs more than stem cells. Stem cells need the support of 3 essential components to get optimal and consistent results. Number 1, growth factors. Growth factors activate and stimulate the stem cells and put them to work. Number 2, bioactive molecules such as cytokines, HA, hyaluronic acid, collagens type 3, 4, etc. This is the brick and the mortar. These are the tools that the stem cells need to do the repair work. And number 3, scaffolding. Scaffolding is the structure or the framework from which the stem cells can rebuild, renew, and re-engineer connective tissue. Now, as you can see, stem cells are crucial, but without the other 3 essential components, the results of stem cell therapy may be disappointing. When we source stem cells from your own body, for example from the bone marrow of your hip, and then relocate those stem cells to the injured area, for example your left arthritic knee, all we are doing is simply relocating your own stem cells, but we are really not adding anything to that treatment. So the essential components of tissue engineering may all be missing, such as your growth factors, your bioactive molecules, and your scaffolding. So the question becomes, why source your own stem cells? First of all, they are very low in quantity and quality, and second of all, the essential components for tissue engineering may be missing. Reason number 4 not to use your own stem cells. It requires a surgical intervention. Mostly, stem cells are obtained from your adipose tissue through liposuction, or stem cells are extracted from the bone marrow, usually the hip. Now, these 2 surgical interventions not only may be painful, but also pose an unnecessary risk to the patient and liability to the healthcare professional. Reason number 5. Stem cell-based therapies that use your own stem cells lack uniformity and quality control. The adult stem cells are usually derived from bone marrow or fat cells of the patient him or herself, which are extracted in one procedure, isolated, treated, and amplified, and then reinserted to the target pathological area through another interventional procedure. There exists a wide variety of methods by which the cells are isolated, treated, and amplified. And these methods can vary even from procedure to procedure within a single clinic. Most of these processes are proprietary and many are protected by patent. As a result, there is no simple way to determine if a procedure or a particular application has any clinical validity. Much of the information available takes on an air of being anecdotal and resistant to investigation through rigorous scientific method. Most, if not all, the current clinical use of adult stem cells in the United States would fall under this rather inauspicious descriptor. Conclusion. We recommend you do not use your own stem cells, and here are the 5 main reasons. Number 1, the quantity or the amount of stem cells in our body drastically declines with age. Number 2, the quality of our stem cells in our body also drastically declines with age, which means that their ability to replicate and differentiate into specialized cell types drastically reduces. Number 3, the essential components for proper tissue engineering may be missing, including growth factors, bioactive molecules such as cytokines and HA, and scaffolding. Number 4, this use of adult stem cells requires an unnecessary surgical intervention which increases the risk to the patient and the liability to the healthcare professional. And number 5, this procedure is not standardized, lacks uniformity, and there's no quality control. Quite frankly, the use of adult stem cells is outdated and is far inferior and has many disadvantages to the use of the Wharton's jelly of the umbilical cord tissue.